Some years ago, I was flown by British television to Washington because there was an emergency meeting taking place there that a monkey virus was being found in human cancers, and they thought it probably came from a vaccine. After attending this meeting, I was my co-producer and myself. There was only one other journalist that sat through all the meetings. Everyone else went home after the press conference. And when I came back to England, I organised searches in the archives. Well, over the next few years, I kept on searching for more documents, and I found they had follow-up meetings at the NIH, that's the National Institutes of Health, the biggest scientific institution in the United States. And I found there had been meetings between the scientists of the UK and the United States, the vaccine safety scientists, the ones that are responsible for the health of our children. What I found were documents that were recorded transcripts, no journalists present in the room. They're then filed away with no note of on what they're about. So you have to read through lots of documents. But this is what I found. I found descriptions of how they made the vaccines. The measles virus, just the same as the mumps virus, same as the yellow fever virus, is grown on the embryos of birds. We basically get fertilized eggs, wait till there's an embryo, kill the embryos, mash them up, and put them in what they call an incubator. They then wait 2 or 3 days, and they add some previous culture, similar culture to it, and then they pipe off the fluid from underneath the cell culture to be made into the vaccine. They filter out everything bigger than a virus. So they filter out bacteria. But they can't filter out viruses because they want measles virus, mumps virus in the vaccine. And what these scientists, named scientists at our senior establishments have reported, this means there could be prions, mad cow disease could be in there. There could be oncogenes. They say this. We don't know if they're there. There's all sorts of cellular debris from the dead birds in the vaccines. Do we filter this out? No, we don't. Have we purified the measles virus? No, all the live virus vaccines are minimally purified. Quote unquote, they're saying that. So a vaccine contains every normal licensed vaccine, every one of the vaccines our children are getting, contains fragments of DNA, fragments of RNA, genetic codes that these scientists report are highly mutagenic, and they say at these meetings, well, they could cause autoimmune diseases and cancers, and they've got no reason to say they don't. They simply say we don't know what they're causing. The chairman of the meeting said, well, have we tested it for the amount of free DNA from birds? And they said, no, we haven't tested it. We have got no way of testing it. This is before they add aluminum, and it's before they add mercury or any other adjuvants. This is what all vaccines, they call it the substrate. And one scientist from Britain said, if the Greens in my country heard about what we are discussing, they are demanding immediately the ending of all vaccination, because we simply don't know what's in our vaccines. I mean, it's quite foul how they make the measles. I then went to, only last year, I went to the records of the Centers for Disease Control in Washington, a major research laboratories, to find out how they isolate the measles virus. And I didn't expect anything like HIV, because surely this is one error. There is a paper So I've uncovered at the CDC, and it's called isolation of measles virus. It says if we think a patient might have measles, got spots, get some fluid from that patient, a sample of their urine or a swab from the mouth.
Start from where the measles have spots. You rub your
I'm sorry.
Sorry.
Start from where, sorry?
From the spots. Red spots. The measles.
So I'm not quite with you, sorry.
Start where you say, check the symptoms if they have spots.
Check the symptoms of the patients. If these patients have got spots, if it looks like they might have measles, but you want to prove they got the measles virus in them and isolate that measles virus, what you do is you take some fluid from the patient and put it in the fridge. This could be some urine, it could be a swab from the mouth with some saliva. So that's what you've got to test. But all viruses are produced by cells. So the next thing you've got to do is get a marmoset monkey. If anyone's been to the zoo and seen the marmoset monkeys, they're the ones that are extremely lively and got long tails. Kill it, take its cells, and put the cells in the cell culture, because they're 10,000 times more responsive to measles than our human cells. That's what it says. But the cell culture isn't ready yet to grow measles virus. The next thing to do is to make the monkey cells cancerous by, say, exposing them to radiation. You immortalize them, as it's called. Still not ready? Next thing is you give those monkey cells Epstein-Barr syndrome, which is a horrific disease. You're still not ready? Next thing, you add a toxin to the cell culture that is so dangerous that the advice is wear rubber gloves. Don't let the human skin touch it. The CDC says at this point your cells are starting to fall off the side of the laboratory vessel. In other words, they're poisoned, they've got cancer and Epstein-Barr syndrome, and they're falling over. They're ill. Give them 2 days to recover and add some nutrients. Now, get the sample out of the fridge from the patient and add it to these deceased cancerous cells. Watch with a microscope for 2 days. If after this time 50% of the cells are distorted, then you have got an isolate of measles virus and you're instructed to put it in the fridge and keep it, because it could be used as a vaccine. At no point, you will notice, at no point is a measles virus seen. No point is a measles virus proved to cause the illness in the cells. We know they're poisoned. We know they've been given cancer. We know they've been given Epstein-Barr syndrome. That is now ready to be made into a vaccine to put in our kids. This noxious mixture is a basis that can be used for vaccines. This is current today on the CDC website. They've actually, if you dig deep enough, you'll find these instructions for the lab technicians are in place on their website. I'm sorry, it's nearly speechless, because I mean, we've all been hearing about how bad is HIV for the last 3 hours, but this is what they do with our kids. This is how they isolate the measles virus. It gets a little bit worse, I'm afraid, too. The meeting I got the transcripts of was co-chaired by a representative of the World Health Organization, and she reported to the meeting that chicken leukemia virus is in the measles vaccine, and they haven't been able to remove it. It's in the measles vaccine all over the world, not just the United States. It's widely distributed in the vaccine. They've known about it for 10 years. They've never told the public. They talk about the dangers of avian flu virus. Well, avian leukemia virus to me sounds much more dangerous. They inject it into our kids. So I went to see how long did they safety test the vaccine for signs of cancer. I found out 48 hours. Now we all know cancers take years to develop. Why can't they test for longer than 48 hours? It's said because their cell cultures don't stay alive longer. So these cells that, from the monkeys that have been poisoned, given cancer, die within 2 days. So they can't test to see what's happening to the chicken leukemia virus. I think this is enough evidence to have measles virus withdrawn now. I'm sorry. I've been horrified by what I found.